Good afternoon, everyone. I, Ms. Kartiki Bhandari, once again welcome you all to the second session of 1 week online international e-FDP ESTP on empowering pharmacy education, navigating regulations, global trends, and advancing outcome-based education and research under the aegis of IQSCL in association with APTI. Today, we are honored to have with us our second speaker of the day, Dr. Shital Butani ma'am. Dr. Shital Butani ma'am completed her PhD in pharmaceutical sciences and MPharm in pharmaceutics. She has more than 23 years of experience in teaching, industry, and research. Currently, she is working as an associate professor in the Department of Pharmaceutics, Institute of Pharmacy, Nirma University, Ahmedabad, India. She worked as senior research scientist at F&D Department for Regulatory Market, ANDA filing in US FDA in Cadila Healthcare Private Limited, Ahmedabad. She received Project Excellence Award for one of her formulations during her tenure in Cadila. She has completed major research project worth rupees 21 lakhs from Institute of Plasma Research, Board of Research in Nuclear Sciences, entitled Exploring Non-Equilibrium Atmospheric Plasma for Effective Sterilization, including biological safety aspects, as principal investigator. She is also working on another project worth rupees 33 lakhs entitled Feasibility Study of wastewater treatment using non-thermal plasma as principal investigator, granted from the Board of Research in Nuclear Sciences, Department of Atomic Energy, Government of India. She has developed nasal and oral spray for an Indian industry which is available in market with the name Erasavir. She has received the Best Paper Award from Indian Drug Manufacturers Association for one of her publications. She has received a minor research grant from Gujarat Council on Science and Technology worth rupees 3,60,000. She also received a travel grant from DST India for attending a conference in Lisbon, Portugal. She was felicitated in the year 2014, 2015, and 2021 during Foundation Day at Institute of Pharmacy, Nirma University. She was also a national volleyball player in a junior team. She has published more than 60 papers in reputed international and national journals, presented more than 35 papers in various national and international seminars. She has guided more than 35 MPharm students and 5 PhD students. Her total citations are 1,743, H index of 22 and I-10 index of 29 on Google Scholar. Her area of research includes solubility enhancement, pulmonary drug delivery system, controlled release formulations, nanoparticulate drug delivery systems using QBD and DOE. Today, ma'am is going to guide us on pharmaceutical product development, a quality by design perspective. I welcome you, ma'am, and without further ado, I request you to hand over the session.
Thank you for nice introduction. Am I audible?
Yes, ma'am, you are audible.
Hello. Okay.
Yes,
Yeah.
ma'am.
Thank you. I would like to thank organizer for inviting me to deliver a talk to faculty and students. Today, I'll be talking about quality by design, various steps, how actually the quality by design can be implemented. First of all, I would like to show you the campus of Nirma University. So I'm working with Nirma University in Gujarat, Ahmedabad, Gujarat. So we also have a very nice campus over here, and I invite all of you to come and visit us. Especially in January, every year we organize a conference. Like this year, it will be NIPICON in 2026. So I invite all of you to come and attend. Starting with the my talk, first I would like to quote here the point which is published in the Wall Street Journal in 2003. What they wrote is pharmaceutical manufacturing techniques lag behind those of potato chips and laundry soap makers. So at that time they were quoted that we are not following a good approach while we develop a formulation. Even during manufacturing also, we are lagging behind potato chips, the example they have given. So let's take an example of potato chips itself. Let's take an example of Lay's. Okay, so every time when we open a Lay's packet, it should taste same. So how they are maintaining that? So as to maintain that quality, otherwise customers may shift to other options available. So if they want to maintain the same quality in the product, they need to follow some standards. So starting from purchase of potatoes. So what this Lay's industry is doing, they do hire farmers. They provide seeds for growing potatoes. They also give the guideline how potatoes should be grown, and they should be of specific size, specific category. There are different categories, varieties are available in potatoes. So specific category they are using. Then once the potatoes are grown, they may check it for quality like starch content, water content, etc. And then selected potatoes are used. Then another material required for manufacturing chips may be oil. We need to fry it in different oils. So again, they have a very stringent specification that this quality oil is required. The specific gravity should be like this. The rancidification must not be there. And so again, the list of evolution tests are there so as to select the specific oil for frying. Then they sprinkle the masala on different chips. So that masala, again, the particles of each and every ingredient which is used to make a masala should be uniform every time. They should be mixed in a proper proportion. So all of this we can consider as a raw material for manufacturing of potato chips. So they have a good control on raw materials. Then another point is process parameters. Now how they are processing that potatoes so as to convert into chips. So they need to wash or clean the potatoes. Then it should be peeled off. Then it should be cut into the specific thickness slices. Then it should be fried at a specific temperature for specific time. So all this process they might have optimized. Like let's say it is fried in two different pans. So in first pan they maintain the temperature, let's say 60 degree, and frying is done for 10 seconds. Then the chips are transferred to another container where temperature is different. So they have optimized the process. What I want to say is, okay, so as to get the same quality potato chips. Even when they sprinkle masala, it should be sprinkled in a way that every chip is exposed. Okay, so that's why the taste, similar taste is there. So what I want to say is this potato chips manufacturer, they have a good control on whatever raw material they are using, as well as good control on the process. So that is required in pharmaceutical industry. We can even take another example of car manufacturer. So what car manufacturers are doing? They are not manufacturing each and every spare part on their own. They may buy some spare part from some XYZ industry and the other spare part from other. So now if they spare part, each and every spare part is not exactly as per the specification, there may be, let's say, if variation is there in a bolt or any other part by 1 mm, then it may not fix with each other, and that kind of car may not run on the road. So what is required? Good control on raw material, whatever material we are using for development of a product, and what process we are following. So QBD is nothing but it is an approach which can be used so as to systematize each and every parameters which might impact quality of my product. We are building quality in a product. So I'll be covering all this point in my presentation. So first we need to understand what is quality. So in this example, we have so many balls over here. And if I ask you to select a quality ball, how you will select? If we want to play a basketball, we may select this one. If we want to play volleyball, we need to select the other one. If we want to play cricket, this ball is a quality ball. We cannot play cricket with the basketball, isn't it? So what is quality then? So quality can be said as fitness for use. So whatever is the application, whatever is the use, it should be fit for that purpose, and we can call that as a quality product. So in pharmacy, if we want to define what is pharmaceutical quality, we can say that if the products are free of contamination and reproducibly, it delivers the therapeutic benefit which we have promised in the label to the consumers, then we can call the product as a quality product. Now how we are testing quality, that whether product is quality or not? So there are different in vivo and in vitro performance tests which we perform, and then ultimately we find out that whether the product is quality product or not. So first of all, I'll explain that how formulations were developed in pharmaceutical industry when I was working with Zydus. So we used to use a univariate approach. That means one factor we may vary at a time. So let me give you an example. Let's say if I am developing a generic version of venlafaxine hydrochloride, and the RLD reference formulation or a brand formulation, that is Effexor XR. So I just want to develop a generic version of Effexor XR. So what we were doing, like we may take the API, we may select one or two diluent, we may select one or two binder, and then initially we may prepare one batch, and then we characterize that batch for its disintegration, for dissolution. Then if we find that okay, the disintegration time is high, we may add some disintegrating agent, take another batch. Then one after other we were changing one of the ingredients, and that is how we are coming to final formulations where we may observe the similar parameters, the quality parameters, that is disintegration, dissolution, to that of the RLD, and then we may go for bioequivalence testing. In bioequivalence it may pass, it may not pass. If it passes, we file the ANDA to US FDA, and then ultimately if we receive the approval, we may market the product. So this is called an univariate approach. One factor we are varying at a time. So what we were doing in this univariate approach, we are checking the quality. We are just testing the quality of end product, and we are not building the quality. Let's say if the product fails in any of the quality parameter, what to do? Once we got approval, we are now going for commercial manufacturing, and let's say the tablet fails into a disintegration test. Now what to do? Are we going to throw that batch, or we will be compromised with the quality of my product? It's a big question. Okay, so generally end product testing we used to do, which is an empirical or observation based approach. It is not well planned approach. The risk is also there of releasing poor quality product if the quality by design approach is not used. We don't have much learning. The learning was very little, although we were used to do the validation. Then it is labor intensive and inefficient approach. And if we want to change something after the product is we got product approval, then we don't have any justification so as to consider that that change is not going to affect quality of my product. But instead, if we use a quality by design approach, we can come out of all these shortcomings of a univariate approach. So let's understand what is quality by design. It is nothing but one approach how one should work. Okay, so it is a systematic, holistic, and proactive approach to pharmaceutical development. Not only pharmaceutical development, I will be using example of formulation development, but this approach can be used for any other method development or any other place it can be used. So what is it? It is an approach to pharmaceutical development which begins with predefined objective. So when you start working, you should have your predefined very clear objectives, that what exactly I want to do. It emphasizes on product and process understanding. So we need to understand that what is my product, what quality I want in my product, what process I will be using. Each and every parameter of process I need to control. So first we need to understand product and process, and then process control should be done based on sound scientific and quality risk management. We will understand it well when we'll go through the example of QBD. So, why QBD is important? Why we should use QBD? It gives high level of assurance of product quality. There are no chance of product failure. Once you develop the formulation using QBD, you have understood each and every important, or generally the terminology used as per QBD is critical. Each and every critical parameter well, whether it is a raw material related, whether it is a process related, so chances of failure are least. There is a cost saving and efficient approach. Previously industry was thinking that there are so many trials unnecessary, are required as per QBD, and so it is not cost effective. But later on, they understood that there is a reduction in product recalls, product rejects, and product recalls from market. And ultimately, they found that the using QBD is a cost-saving approach. It increased the manufacturing efficiency, it reduced the cost and product rejects. It minimized regulatory non-compliance issues. So you may not get so many issues or penalties from regulatory bodies. It enhanced chances of product approval in first attempt. So if you have used QBD, there are no loopholes left, and so you will straight away get approval of your product. It streamlined post-approval manufacturing changes and regulatory process. You will have a justification that if you make this much change, it is not going to affect my product quality because you have studied this while you have developed the product. And so post-approval changes are easy. We have opportunities for continuous improvement if we are using QBD approach. So ultimately, in conclusion, what QBD is doing, it reduces variability, and that is how it increase the quality and reduce the cost of product development. I'll suggest all of you, and I will also be referring this guidance document of US FDA for understanding what is QBD. So it is quality by design for ANDAs, an example for immediate release dosage form. Such two guidance documents are there. Another is for sustained release formulation. We will be referring today about immediate release dosage form. So these are 7 components of QBD, or rather we can say 7 steps how we should proceed for applying QBD in our project. So the first step, that is quality target product profile. We need to define what is my target. Quality target product profile. So starting with this, let's say what is it? It is a summary of quality characteristics of drug product. So just we need to summarize that what exactly I want. Like let's say which dosage form I am developing. So if I am developing a tablet dosage form, why tablet dosage form? Why not any other dosage form? So dosage form we need to finalize which strength we will be using. So we will see each of this in detail. So first, if we go through the definition, it is a prospective summary of the quality characteristics of a drug product that ideally will be achieved to ensure the desired quality, taking into account safety and efficacy of drug product. So all of this we need to consider. So in this guidance document, we will take an example of developing an ANDA product for acetylptan 20 milligram formulation. So we will understand what is it. So we can see that these are all quality elements. Here in this column, all quality elements are mentioned. So whatever dosage form, this example is for tablet dosage form, but whatever dosage form you are developing, you can apply the same concept. So the first is dosage form, as I said. So I am preparing a tablet. Why tablet? Because right now this is an ANDA formulation development. So I want to prepare a bioequivalence formulation to that ANDA product, which should be pharmaceutically equivalent. So I can justify that the formulation I am developing is a bioequivalence formulation. And so it has to be pharmaceutical. Because it is pharmaceutically equivalent product, I need to prepare same dosage form. So it is a tablet. Then what is dosage design? I'll be preparing IR tablet, immediate release tablet, because again I want to match the bioequivalence with IR tablet. So I need to prepare it, IR tablet, without score line and coating, because the RLD doesn't have a score line as well as coating. So I will also not go for that. Then route of administration. So it will be oral. Dosage strength, it will be 20 milligram, because again I am developing a pharmaceutically equivalent product. What should be my pharmacokinetic requirement? So the RLD shows that Tmax is 2.5 hours or less. So again, I want the same Tmax. I want that my formulation should be bioequivalent to that. Stability, I may target for 24 months minimum, or better than that is okay. Then drug product quality attributes. So formulation I'll be evaluating for its physical attributes, identification of API, assay, content uniformity, dissolution, degradation. Here the list may vary depending on which dosage form you are preparing. Then accordingly, here also, so as to maintain the standards, I need to meet the all these important criteria. Then similarly for container closure system and so on. Okay, so this is nothing but QTPP. So first step is you need to finalize what exactly you want. Okay, now the next step to that is identifying critical quality attributes. So critical quality attributes are nothing but the important evolution parameters. There may be all evolution parameters, but out of that, what are critical, what are important, that we need to find out. So let's see about critical quality attributes. So it is a physical, it can be of any in nature. Okay, it can be a physical parameter, chemical parameter, biological parameter, microbiological property, anything, or any other characteristics that should be within an appropriate limit or a range or distribution to ensure desired product quality. So these are critical parameters we will be identifying out of all parameters. So identification of CQAs from QTTP is based on severity of harm. How harmful this, if let's say dissolution. So if it doesn't meet dissolution, then it may harm the patient. It may affect the efficacy. It may affect the safety. So severity is high. So dissolution should be a CQA. So likewise, we will see examples. So this is a table given in the same guidance document. Let's say we are considering the physical parameters first. So appearance. So appearance is not critical. Here in this column, you can see that whether it is a critical or it is not critical. So no and yes is mentioned. So for any dosage form, you can prepare this table with justification. So appearance of a tablet. It is important from patient point of view that it should not be broken. It should be it gives some psychological impact on patient minds. Okay, so it should be good in appearance, but the appearance is not going to affect the safety and efficacy of my product. It is not going to affect safety and efficacy. So that's why it is not critical. I will have a good appearance. There may not be any visual defects on tablet, but it is not critical. So it is not my CQA. Similarly, odor, size of tablet, then score for configurations, friability. Even friability, definitely I'll be keeping friability as per USP requirement, that is less than 1%. So it may not be critical. Now identification of API. So definitely it has to be acetyltryptan API. So it is critical. We cannot have any other API other than acetyltryptan. But here asterisk mark is there. So it is critical, but it is not being affected by any of the formulation or a process parameter. Will it? કે હું ફોર્મ્યુલેશનમાં આ એક્સિપિયન્ટ વાપરું છું તો મારું એપીઆઈ ચેન્જ થઈ ગયું એવું તો થવાનું નથી. It is not going to happen. है ना? So as API has to be acetyltryptan, it is very critical, affecting safety and efficacy. But I did not need to consider this while I develop my formulation, while I develop my process. So it is critical, yes, but it will not be considered in every step of formulation development. Then assay of tablet has to be 100% or whatever is the limit. Within that limit, it is critical. Yes, assay may affect the safety and efficacy of my product, so it is critical. Similarly, content uniformity, dissolution. So these are the requirement. My targets are there as per my reference listed product. So content uniformity should be performed as per USP, and it should pass the acceptance criteria, acceptance value less than 15. Then dissolution. Yes, dissolution, whatever is mentioned, like 80% within 30 minutes, then my formulation must have dissolution 80% within 30 minutes in whatever dissolution media and conditions suggested. So this is my CQA. So I hope you understood what is CQA. These are quality attributes which are critical. Likewise, there may be so many other examples, like we can have a weather degradation product, whether it is critical or not, then residual solvents. So residual solvents are also critical. It may affect the safety. There is a guideline of residual solvent. And so the residual solvents should be less than that. But I do not need to test my every batch while I am developing a formulation for residual solvent. So it is critical, but a strict mark is there that I will check for final product, that it will not be, the residual solvent must not be there in my final product outside the limit specified in the guidance document. Water content is important, but my product is not hygroscopic. It is a solid dosage form. So microbes may not grow, and so microbes must not be there in the formulation, but it is not critical for this purpose. So this is how CQAs are defined. So what we have done is, we have first of all prepare a quality target product profile. Followed by that, we have identified what are critical attributes, quality attributes. Now next is, I will be starting formulation development. So I need to consider critical material attributes and process attributes. So let's see that. So first of all, I need to understand that what all parameters are going to affect my CQAs. Okay, so I have identified my CQAs: dissolution, assay, content uniformity, and degradation product. So all these 4 CQAs are being affected by what? The people working, the environment might impact the CQAs, the measurement I do, the process I use, the material I use, the equipment I use, and there may be other than that also. So what all parameters? First, I need to prepare a list of parameters which may impact CQA. So generally, this is called Ishikawa diagram, or it is also known as fishbone diagram because it looks like a fishbone structure. So here is an example of Ishikawa diagram. So if I am preparing a tablet, the raw material, the properties of raw material may impact. It includes API and other excipients. The granulation process, let's say I'm using a granulation process. So all parameters of granulation process, the drying process, the analytical method I am using, the compression process, the other factors of environment of plant, all of this may affect my tablet quality. So likewise, first of all, we need to prepare an exhaustive list of factors which may impact, and then we need to identify how much this parameter has an impact on CQAs. So that is done by using another next step, that is the initial risk assessment. So each of API characteristics or the parameters, the excipients related factors, and the process related factors, for that I need to prepare initial risk assessment. I need to do that. So what is initial risk assessment? So based on my knowledge, I can define that whether I can define the impact of each of these process parameters or formulation parameters onto my CQAs. So whether we will take first example of drug substance. Let's say the drug substance is a API. So we will see that how the API related attributes affect the CQAs. So we can categorize this into 3. The API property which may have a low impact, either medium impact, or a high impact on my CQAs. So when I can say low impact, if there is it is acceptable. If it is not going to have a much impact, I do not need to further investigate this parameter, then I may write low. When I can write medium, the risk is acceptable, but still some investigation need to done so as to reduce the risk. Then I can write medium impact, and when I can write high impact, the risk is unacceptable. If there is a variation in this parameter, the quality might be affected. So what I need to do is, I need to optimize. I need to do lots of investigation so as to reduce the risk of this parameter. So let's take an example and understand. So here is the critical material attributes related to API, and I am doing a initial risk assessment. Just I am assessing how much risky this parameter is. So let's take first example of solid state form. So if the drug is existing in more than one polymorphic form, whether that polymorphic form is going to affect my assay? No, not really. If polymorphic form is change, or if I use a different polymorphic form, do you think because of that assay is impacted? No. So I can write low impact on polymorphic form on assay. Similarly, it is not going to affect content uniformity. But different polymorphic form may have a different solubility, and so their dissolution might vary. So whatever polymorphic form I am using, I should check it for its impact on dissolution. It may impact degradation because the crystalline structure may change, and because that structure may change, it may get differently affected by different parameters. So there may be a little chance of its impact on degradation. Let's take another parameter, particle size distribution of my API. Will it affect assay? It can affect, but not to the extent it affects content uniformity. So the particle size distribution is acetyltryptan example I am taking, and this API is poorly soluble. So when it is poorly soluble, it impacts the dissolution to high impact. Particle size may impact the dissolution. So we need to write high over here. Particle size is not going to impact degradation. So for each CQA, what I have identified, I need to see that how much is the impact of different drug substance attribute on this CQAs. And what I need to do is I need to optimize all those CQA, all those attributes which are highly impacting my CQA. I hope this is clear to everyone how risk assessment is done, initial risk assessment. And here you need to justify. So as we have discussed that for solid state form, in assay and content uniformity, we have written that drug substance solid state form does not impact or affect tablet assay and content uniformity. So here risk is low. So I can write low in that. But the dissolution, different polymorphic forms of drug substance have a different solubility, and it can impact tablet dissolution. Risk is high. So which polymorphic form I'll be using in this project? Polymorphic form 3 is used. And so 3 should remain. The polymorphic form should not change while I develop the product. If it change, it may highly impact the dissolution. So I should be careful. Likewise, for each and every drug API attributes, how these API attributes are impacting my CQAs, I need to write a justification why I have written low, why I have written medium, why I am writing high. So I have done the initial risk assessment for API. Similarly, for excipients, the critical material attributes of related to excipients. So let's say I'm developing now formulation. So in formulation, I am using microcrystalline cellulose and lactose in different ratio. I am using cross-camelose sodium, talc, and magnesium stearate. And I just want to understand how these are impacting and particle size of API we have already discussed. So how they are impacting my CQAs. So the ratio of MCC and lactose may impact assay to a little extent, but may also impact to a higher extent to content uniformity, because if particle size may be different, density of both of these diluent may be different, and so it may affect content uniformity. One is soluble, one is insoluble, so it may affect dissolution if we change the ratio, but it may not affect degradation. We have done some pre-formulation studies with this acetyltryptan, and we have observed that it is compatible with MCC as well as lactose, and so I can write low in degradation product. Similarly for all other parameters. So here for magnesium stearate, when we have done pre-formulation studies, we have observed that magnesium stearate may affect the degradation of API. So that's why I have written medium over here. And we all know that magnesium stearate is highly hydrophobic API, so it may affect the dissolution. So such justification should be given that why I am writing low, why I am writing medium, why I am writing high for any of the formulation related parameter. So this is initial risk assessment of critical material attributes related to excipients. I'll skip this now. You can go through this guidance document later on. Similarly for process parameters. So here I am developing a tablet using dry granulation process. And so for that process, what are the process steps and how they impact my CQAs? Similarly, I need to do the initial risk assessment and with justification. So what we have learned till now, we have seen that what is QTPP, then we have understood what are critical quality attributes. We need to identify the evolution parameters which are critical. Then we need to identify API properties or the excipients related properties or process parameters which are critically impacting my CQAs. Based on that, I have done the initial risk assessment. So now I know that which is critical and which is not. So what I need to do is I need to optimize all critical attributes. And optimization generally is done by using design of experiments. So we all know we might have used design of experiment. So what are the key component of design? It can be we need to identify factors. So already by doing initial risk assessment, we have identified factors and their levels. So let's take an example that if we are using a MCC to lactose ratio, so this is one of my factor. And their level is either I am using 60 is to 40 and the other another level is 40 is to 60. So I might be evaluating two levels. And the responses are nothing but my CQAs, critical quality attributes. So various designs you might be aware about, like their designs can be divided into either screening design or it can be optimization design. If it is a screening design, if we have so many factors to be characterized and just we want to understand whether this is important, this factor is important or not. So sometimes it happens, like let's say I have never developed liposomes and first time I am developing liposomes. I may not have that basic knowledge whether this factor is important or not. So I may go for screening design. Screening design will tell you whether this factor is important or not. It is not going to tell you about the impact of this factor on CQA, how much it impact, or whether the interaction is there between two factors or not. So that kind of information we may not get from screening design. So Plackett and Berman design and fractional factorial design are widely used to screen the formulation. The other designs are optimization design. So if I'm aware about, like these are the two important factors which are going to impact my formulation. So then I can go for optimization where I will get better information compared to just screening. So here I'll get information about whether the interaction is there between two factors or not, three factors or not, and so on. So preferably I may go for factorial design, I may use central composite design or Box-Behenken design in pharmacy, or sometime if I am developing emulsion type of formulations where I can use mixture designs. So you need to select the experimental design wisely. What is your purpose? What is your objective? You need to understand, and based on that, and how many factors are there? Based on that, we can select the suitable experimental design. This is an example of outcome of experimental design. So why we are using design? Why not we go for one factor at a time? Variation, okay, univariate approach. Instead of that, here, for example, I am studying the effect of inlet air temperature and the effect of sodium caprate as a permeability enhancers or solubility enhancers on solubility and permeability. And this is my outcome overlay curve I hope so. So what this overlay is saying, if I use sodium caprate in this range, yellow part range, and if I use the inlet air temperature in this yellow part range, I'll get my solubility and permeability as per my requirement. So in future, let's say this is my optimized batch I have selected. sodium caprate concentration is this, and I'm using the inlet air temperature at this. Okay, but in future, by mistake or intentionally, if I want to change the inlet air temperature to let's say from this to this, or maybe from this to this, I can justify because it is still within my yellow region. That means it is acceptable to regulatory agency. My solubility and my permeability are within the limit. It is not going to impact. If this change is there, it is not going to impact my solubility and permeability. So regulatory agency may accept this change. So what is QBD? It is a systematic approach, and ultimately we have some data with us which are statistically treated, and we know that how much variation is acceptable and how much variation is not acceptable. So we have done optimization for each material attributes and process attribute. We need to do that for each material attribute, for each process attribute, that how it impact my CQS. So once optimization is done, it is a very exhaustive process. So once optimization is done, I can prepare updated risk assessment. So it is just like a risk assessment, initial risk assessment. Now we need to update. So what we are doing, initially when we have written high or medium, now the risk becomes low. How risk became low? Because I have critically controlled this process variable. I have set some range for this. Like let's take an example of particle size. In this guidance document, they said that if we are using a particle size D90 less than 30 micron, then the resolution, the bigger is acceptable. But if I use particle size of API beyond 30 micron, D90 beyond 30 micron, then the resolution may not as per my requirement. So I have set the limit for particle size, and that is how now risk is low. Similarly, I've set the ratio of MCC and lactose, and so now risk is low on SA, content uniformity, degradation. So we can prepare the updated risk assessment where now risk is reduced to low. How it is reduced to low? Because I have optimized the impact of this on my CQS. And if I use within the identified range all these excipients, then all of this requirement will be fulfilled. So what I'm doing is I'm building quality in my product. So we can prepare an updated risk assessment with justification. In the next step of quality by design, that is control strategy. If anyone has any questions, they can ask in between anytime. So what we have done, we have identified quality target product profile, then critical quality attributes, then we have identified critical material related, which is API as well as excipient, and process related attributes and its impact we have studied. First of all, we have identified whether it is in a risk assessment, whether all of these have a high impact, medium impact, or low impact on my CQS. That I have done during initial risk assessment. One, I have identified those who have high risk. I need to optimize them critically. Once I have done optimization, or once I have studied all that risky parameters critically and I have fixed the range for all the parameters, now my risk is reduced to low. So I prepare updated risk assessment where the risk is low. Now I'll be preparing control strategy. So what is control strategy? The control strategy is a planned set of controls derived from current product and process understanding that assures process performance and product quality. I'm repeating it, a planned set of controls. What is required? Now I have studied effect of particle size on my old CQS, and I have now control on particle size. So if I control particle size within that range, Now I'm assuring the performance of my product, the quality of my product will not compromise. Okay, so this is one example of particle size. There are so many other parameters which are controlled now. So it is a planned set of controls that is derived from the current product and process understanding that assures that process performance and product quality is as per the requirement. The controls can include parameters and attributes related to drug substance, drug product materials, components, facility, equipment, operating conditions, in process controls, finished product specification, anything. So now everything I am controlling well so that my product will be a quality product. So here is an example of again same acetiptan. So what I have studied? I have studied the acetiptan should be, it exists in different polymorphic form, but I will be using polymorphic form 3. How I will assure that it is polymorphic form 3 only? So I have studied that 2 theta, this is generally done by using XRD. Okay, so if I am using XRD, sorry, at a specific 2 theta value, should be there. So peak should be there at this 2 theta value, that is 7.9 degree, 12.4, 19.1 and 25.2. So whenever I will procure my API, I will be analyzing API for by XRD for this peak at this 4 critical 2 theta value. There should be a peak as per requirement. Then it ensure that polymorphic form is form 3. Same way, if we consider particle size distribution, I have studied D90 starting from 10 to 45 micron. A big range I have studied. Same for D50 and D10. But what I have selected the range, proposed range for commercial batch. So I am proposing that the particle size D90 has to be within 10 to 30 micron. D50 should be 6 to 25 micron and D10 should be 3.6 to 14.4 micron. So every time when I purchase my API, I'll see that it has to be within this requirement. The particle size of API must be within this requirement so as to get the required dissolution, required in vivo performance, that is the bioequivalence or bioavailability, as well as for batch to batch consistency. Same for all other parameters. So lactose, monohydrate I have used, which grade, then microcrystalline cellulose. So again, particle size of this excipient is going to impact my performance or quality of my product. So what particle size range I have used and what I am proposing. So particle size should be within the used range, 70 to 100 micron for lactose monohydrate. Here is a typographical error. And 80 to 140 micron for microcrystalline cellulose. So again, same particle size every time I should use so as to ensure the sufficient flow property and batch to batch consistency, and so assay and content uniformity will be within the limit. Same for other excipients, particle size. Then similarly, you should have a planned set controls for all process parameters. Like V blender I am using, so at what RPM I will be rotating, how many revolutions, so what is my target. Then similarly, it may affect the mixing, and so it may affect the content uniformity, blend uniformity. So I should control this. Then similarly, how much blender should be filled, whether up to 50% occupancy, whether 35% occupancy, 75% occupancy. So it also affects the mixing uniformity. So I have said that it should be filled up to 67%. That is my control, my target for commercial batch, so that I will have a required blend uniformity. So I hope you understand that what is control strategy. So there may be so many other points in the control strategy for each and every parameter. For the granules particle size distribution, I should fix that it should be within this, it should be within this, other lubrication points, the compression point. So everywhere I can fix that what is my target. This column is very important. Now this is the range I am proposing based on formulation development experiments done for my commercial manufacturing process. And so if I follow all these parameters within this range, definitely I will have a good control on my quality of my product. It will be a quality product. So if we develop a formulation using quality by design approach, we have a better understanding of process, the chances of failure of batch is reduced, we have more efficient and effective control on change if we want to do any change, we have a good return on investment and cost saving, we have an additional opportunity that an enhanced QBD approach to pharmaceutical development will provide opportunities for more flexible regulatory approach. So if you request something to regulatory body and if it is within the range you have specified, then regulatory body may accept it easily. So here is an example. If we are doing some manufacturing changes within the approved design space without further regulatory review, they may approve it. So there is and we may not need to go for post approval changes. It will be reduced like anything. Once we got approval, we have developed the product critically, and so now there may not be requirement of any even post approval submissions. We have a better innovation due to ability to improve process, so there may not be need to resubmit anything to FDA. We have a more efficient technology transfer. It is again very important point in industry. Like many a time it happened that on small scale they may can develop, but then they might have taken only one or two batches on a large scale, and so later on the same technology may not work. It is difficult to go for technology transfer. But if systematically formulation is developed, it is easy to go for technology transfer. That is from small scale to larger scale. We have a greater regulators confidence on our product. The risk-based approach is used, risk is identified and it is assessed and it is control strategy is developed so as to reduce the risk. Innovative process validation approach, we can call it as. We have less intense regulatory oversights and less post-approval submissions. We have a better satisfaction of consumers because of consistency in my product. We have more drug availability and less recalls, improve yield, low cost, less investigation, reduced testing, and that is how using QBD approach is very advantageous for pharmaceutical industry. And now they are using it. Here is another example. Let's say if we want to develop a sterile powder for parenteral application. So briefly I'll explain this. So the dose and everything is defined, the volume of injection and everything, the shelf life. Okay, so I need to define a target product profile, quality target product profile for this injection. Then what will be the next step? I need to prepare the critical material attributes. So what are critical material attributes? That the API should be pure, the type and level of impurity present in API, bioburden of API. Here microbial contamination plays very important role because parenteral formulation has to be sterile. Then what is pyrogen level of API and other excipients? Package material I am using for packaging. Quality of water for injection I am using. Residual solvents level. Solubility specifications for API. Buffer type and buffer concentration I am using for making reconstitution. Type and concentration of preservatives I am using in my formulation. So for parenteral formulation, the materials may be different and so material related requirements may also vary. Similarly, what can be my CQS? Process parameters or CPPs, which may impact the CQS. So sterilization condition will impact my CQA because product has to be sterile. If I am using some filtration process or pre-filtration, the holding time, how long I keep the batch. Like today, if I am manufacturing, tomorrow I am sterilizing, and if I am filling the batch into my containers in after 3 days, then if that 3 days hold time, there is a chance that during that product might get contaminated. So how much hold time is there? Then what are cleaning methods I am using? If it is a freeze dried product, because I am developing a reconstituted powder, so what are the conditions of pressure, time, requirement for freeze drying? All of this might impact my CQA. If we take another example, let's say if I'm developing a sustained release formulation by using some the wax, that is carnauba wax, then what can be done? So here again, I'll show you one example. Like let's say I'm developing it by using hot melt granulation process, and this is my process that the grain agglomerates are formed at some low temperature, maybe ranging from 50 to 90. Cooling should be done, and so on. So then, let's say these are the manufacturing steps. That first of all, dry mixing, then melt granulation, then need to sift the powder. These are the conditions I'm using, like sifting and sizing, and then again blending, then finally lubrication, and then finally compression into a tablet. Okay, so if these are my manufacturing process, then this is how I may prepare a table, like how process parameters might impact my quality attribute. So sieving, what size I am using, so as to sieve the excipients at all this manufacturing step may impact my assay, may impact my content uniformity. Same way, the chopper speed, impeller speed of RNG, whatever I am using, the occupancy, how much it is filled, like 60% filled, 30% filled, or 70%. Generally, any equipment is filled 30 to 70%, not beyond that or not less than 30%. Otherwise, mixing uniformity may not get. So, it may impact my content uniformity. It may impact my dissolution. So, likewise, you can prepare a detailed the flow chart, and finally you can optimize them using specific design. Here, let's say it is a three factors are selected, and 2 raised to 3 full factor design was used, and the batches are prepared and controlled. Similarly, let's say if process parameters are there, I may study the RPM, the temperature, the hardness of formula hardness, so as to the force applied so as to get required hardness, impact of hardness on my formulation performance. These are the references I can suggest you to use. Even we have published one book, and QBD is one of the chapter in that book, an essential guide in pharmaceutics. This book is very useful for B.Pharm students, those who are preparing for GPAT, because we have included the chapters which are very much relevant to GPAT, and lots of question and answers, MCQ type of or single line answers are given in that. As I said, you can refer this IR dosage form guidance document, which is very important to understand this. There are other guidance documents like Q8, Q9, Q10, but they are difficult to digest. So those who are studying this for the first time, if they understand it by using this guidance document where one example is been given, it is easy for all of you to understand. I would like to acknowledge Nirma University, late Professor Dr. Mukesh Gokhale, my PhD guide, who has taught me various aspects of pharmaceutics, my colleagues and my students. Thank you, everyone. Now my session is open for question and answer.
Thank you, ma'am. The session is open for discussion. I request delegates, if you have any questions, please write it in the chat box. Okay. Ma'am, we have one question. How do regulatory agencies like FDA and EMA evaluate QBD implementations in pharmaceutical submissions?
They do, like the regulatory body, they do check that whether you have used quality by design approach or not. Okay, so the FDA, they see that see industry, what industry would like to do, they need to try to reduce the development trials, you know. So they try to justify that in most of the factors the impact on CQAs is low, because they may have used that excipients. Let's take an example like MCC, they might have used in so many formulations, so they are aware about like what properties are being affected by microcrystalline cellulose. So they may feel that it is not that important, So they may try to write low, but at least they need to identify minimum required critical parameters and optimize them. Then only the FDA may consider that you have used the uh, approach of QBD. So they do check. They do check. You need to submit all this information in dossier with justification. So definitely QBD, whether it is implemented or not, it is being checked by regulatory agencies. They may have a trust, more trust on you if you have used QBD.
Ma'am, one more question is there. How many critical quality attributes should be considered in any research work?
Yes, so it depends on dosage form. Like in this example of tablet, as I said, like dissolution, assay, content uniformity, and degradation all are critical. Okay, but when we are doing in a institute, sometimes students have a limitation. Like degradation product, for identification of that, you need to have your HPLC method developed. है ना? So if the student is working in for PhD, let's say, then they can try. But if the student is working in M-Pharm, it is okay if they won't consider degradation product as a critical, although it is critical. Okay, but they may have some limitation. So but to understand the approach, and when you complete your study while you are working in industry, it is very much helpful for you to implement this approach over there. So you should have a proper understanding. It's okay if you take one or two parameters more or less while you are studying. Okay, but the concept you should understand that if I'm working in industry, I cannot ignore degradation product. I hope this suffice your query.
Okay, I think there are no more questions, ma'am. I will proceed further.
Thank you.
So, yes. On behalf of Gokhale Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, I express my heartfelt gratitude to our expert speaker of today's second session of e-FDP and e-SDP. Dr. Shital Butani ma'am spared time from her busy schedule to grace this program. Ma'am, your expertise and thought-provoking ideas have truly inspired us. Ma'am explained interesting example of potato chips to highlight the correlation and importance of QBD in optimizing the process parameters in pharmaceutical manufacturing techniques. She also emphasized on what is quality, what is problem identification, what is quality by design, why it is important, and in-depth explanation of 7 steps of quality by design. Ma'am, we are grateful for the time and efforts you dedicated to share your knowledge with us. On behalf of Gokhale Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, I request you to accept the certificate of appreciation presented as a token of gratitude for your insightful session. Thank you once again, ma'am.
Thank you. Thank you. It was very nice delivering this talk. I also thank the organizer for inviting me, and I hope we will have a good interaction in future. Thank you, everyone.
Yes, thank you, ma'am. Thank
Thank
you.
you. Now I take your leave.
Yes, ma'am. Yes.
Okay.
With this, yes, with this, I officially conclude today's session. Thank you all for being the part of this session, and delegates are requested to fill the feedback form. The link of the same will be shared on the WhatsApp group, and I would like to request all the delegates to ensure that they fill their names, affiliation, and other necessary details correctly in the feedback form. This is important for us to process and issue your certificates accurately. Thank you once again.